So very good evening to all the students. After a long break, we are going to. So, as you are aware that in series we invite eminent speaker to speak on various topics related to science and address the queries you have in your mind. So today we are fortunate to have with us Dr. Devrata Vishwas, who is going to speak on understanding of mechanism of gene expression and its implication in combating human health diseases. So I request all our viewers, all our students who are with us in different social media platforms to post their question relevant to today's talk in the chat box. I will take this question with our speaker after his talk. So first of all, let me briefly introduce Dr. Devrata Vishwas, scientist in Molecular Genetic Division of CSIR, Indian Institute of Chemical Biology, which is one of our premier CSIR laboratory. He has done his PhD in experimental pathology from Department of Pathology, University of Utah, Salt Lake, USA, and he has been awarded with various recognitions and awards, including Wellcome Trust, DBT Intermediate Fellow, DBT Ramalingam Re-Entry Fellowship, Leukemia and Lymphoma Society Career Development Fellowship, Mary Jo's and Henry Davis Fellowship. And he is having several publications to his credits. During PhD thesis work, he studied mechanistic study to show the histone protein and which are involved in regulating chromatids. So more will get in detail in his talk only. His current research interest is in the area of understanding eukaryotic transcriptional regulatory mechanisms in relation to the novel factors associated with fusion and fusion partner proteins, study of context-dependent functional regulation of super elongation complex, global transcriptional regulation, study of age-associated functional regulation of complex for understanding age-related human pathogenesis. Introduction. I invite Dr. Devrata Vishwas to deliver today's talk. Over to you, Dr. Devrata. We are waiting for your very fascinating and wonderful talk. Looking forward to you.
Okay, thank you, Dr. Pooja, and good evening to everyone. I hope I am audible clearly to everyone. And what I'm going to start today is that kind of give you a kind of glimpse of, and because the audience over here is fairly, I'm expecting is not, to the level of that we generally talk in the scientific forum. So I would try to put it in a more kind of a layman's language in terms of what we do, and to make you rather more interesting about the things that we are doing in the lab, and then so that some of you perhaps in near future use the biological research as your career for moving forward with your career. Okay, so what we do, and we in IICB for the last several years, what we are doing that we are working on this mechanistic understanding of gene expression and whatever its implication in combating human health diseases. Now, what is gene expression? You must have heard about in these forum, in that forum, or in social media and this and that people do talk about the genes and then its expression and then so many other things. Now, to put you into the context of today's talk, as well as to put you into a very layman's language in terms of genes, and let's just put, because I expect all of you a kind of a school student, so to put you into the context of the gene and its regulation, let me start with a human body itself. Okay, can you go to the next? Yes. As you can see, the human body, I mean, I'm just going to kind of break it up, the whole human body and the regulation into a very, tiniest one that we are working with. All of you know that the human body is made up of several organs, and a lot of it is shown over here, including brain, including heart, liver, lungs, and so on and so forth. And all these organs, they have to work perfectly for proper functions of the human. And there are always checks and balance of these one. Now, going even beyond these, to the next one, no, not this one. Yes, fine. Going beyond even the next one is that the organs that we have in the human body, those organs are actually made up of a tiny units. And few of you may know these tiny units are called cell. So if you, and this in cartoon image over here, it's shown over here that if you just, slice open a cell, what are those organelles that you see within a cell? Okay, as shown over here, and in your biology textbook, you would see this kind of picture of the cells are given in which a lot of these organelles, including nucleus and then mitochondria, the Golgi body, and so on and so forth, all these organelles are there. Now, each organelle has its own function. Okay, for the overall, so within a cell, all these organelles have to function properly, and when all the cells combine together and form an organ, that organ has to perform properly for our everyday function. Now, what we are going to focus for today's talk is the organ called the nucleus. So, if we go to the next slide, then what we see is that within the nucleus, because we are going to talk about the gene, so we'll have to go first to the gene. Where do all these genes reside? These genes reside within the nucleus. Human mitochondria also has some of those genes, but as compared to the nucleus, these number of genes in the mitochondria is far fewer than in the nucleus. So in the nucleus, you see a lot of chromosomes. We humans have 23 pairs of chromosomes, mean 46 chromosomes. Now, within these chromosomes reside the gene or the DNA sequence, which are being shown in the next slide. So, as shown in this one, that if we further break open the chromosome, then what we see that the chromosome, it's a highly compacted structure in which it's once again that can be further broken into a tiny unit called the nucleosomes, which coils with each other to form the supercoiled structure called the chromosome. And within these nucleus, within these tiny units of the nucleosome, you have the DNA wrapped around with a protein called the histones. So the histone proteins with the DNA wrapped around and forms the nucleus that in turn forms the nucleosome that in turn gets supercoiled to form the chromosome, and that's there within the nucleus. And these DNA sequence have to be available for the gene expression. Now, genes are composed of these DNA sequences. So as you can see from, if you break open the human body, where you get ultimately the DNA sequences for the gene expression for any kind of function. Now, when we talk about the gene expression, we always use a basic principle of or the basic central dogma of molecular biology. It's shown in the next slide, which is what it's shown over here. And this is the basic principle of functions of any form of living organism, be it human, be it any other organism that you can think of, a prokaryote, a eukaryote, anything, a lower organism to higher organism. This is the basic molecular biology. The simplified form of the central dogma is that from the DNA, through a process called transcription, we form the RNA is being produced, and from the RNA, through a process called translation, the proteins are being produced. And all the proteins are required for the biological functions. Without proteins, you cannot do any biological function. So whatever you are doing, whenever you are playing, whenever you are doing exercise, whenever you are watching movie, whenever you are studying, everything is being, or the biological functions are being regulated by this process called by these proteins. Okay, and these proteins are being produced from the DNA, which encodes the genes. So from the genes, you get the RNA. From the RNA, you get the proteins, and that ultimately regulates the biological functions, day-to-day biological functions that we do every day. Okay, and then if we go to the next slide, then what we see is that when we carry on our functions and these and that, now every cell has a specific lifetime, and for the cell to grow, the cells die and the new cells are being formed. So cells are being formed from one cell to another by a process called the cell division, or the cells undergo cycle, what we call a cycle. A cell cycle. A cell undergoes, during the division, it undergoes a cycle through which one cell forms two cells, okay, and it has multiple steps of the or the checkpoints or the stages of this cycle, which is shown over here as the G1 is one of these one of the stage of the cell cycle, then S phase, and then G2 phase, and then M phase. Now when the cells undergo cell cycle, in each of these stage, it's perfectly controlled, in a sense that if there are some problem during the stages of this G1 stage of the cell division, then it doesn't go further. Okay, same way, if there is a problem of the S phase wherein the DNA synthesis happens, it doesn't go further. Okay, so these checks and balances have to be done for the proper function of the cells. And if you do not have these checks and balance, then you have a problem, because the cells do not respond to the environmental cue or for the other signal that says it's not the time for you to divide, rather you stop. But when there is a problem, it does not stop, and as is shown in the next slide, that causes this cancer. What is one of the major implications of the study of the gene expression, which I'm coming next in the next few slides, also in some of the current context, but as shown in this cartoon diagram in these cancer development, is that the normal cell, usually when you grow them, and usually we grow in the lab and all these, and when you grow the normal cells and when you give them the growth factors, the normal cell divide from 1 to 2 and then multiple like that. And when you do not give any growth factor to them, it stops growing. Okay. However, the cancer cells, which doesn't really care about all those checks and balances that I talked about in the last slide about during the cell division, it just doesn't care, because it just says, it just thinks that I need to grow. Okay, because the overall regulatory system within the body is, or within that specific cell or within that specific organ, is completely messed up, and because of which it's not sensitive to adding growth factors, and it keeps on growing and ultimately give rise to different kinds of cancer, which are shown in this right panel. And we are not going to go into these types of cancers, but rather going into a little bit more detail about these processes as is shown in the next slide. Okay, at these kind of cancer cell, when they grow, because they do not care about any of these checks and balance, they always keep on growing. And what happens is that at some point, these cancerous cell, which doesn't perform properly, and they outnumber the growth of the normal cell, as is shown in the top panel. And when they outnumber the growth of the normal cell, then the body physiology doesn't really work, and then we have a problem. And then slowly, when the cancer cell goes from one place to another, and ultimately that captures the whole body, and we have a problem, and that the patient succumbs to the overall disease. And if no intervention is there, then obviously the patients ultimately succumb to this overall malfunction of the system. Now the question is, why are we talking about all this cancerous growth? Because, go to the next slide, what we have is that for the continuous production of the cells, or what I say to you guys, that one cell goes to the next one, and in the cancer cell, it just continuously proliferates. It doesn't care about any of the checks and balance and all these things. However, without the help of the system, it doesn't proliferate. So for that, you need to have a continuous production of proteins. Continuous production of proteins are the key biological macromolecules, which in this case proteins, is required for the proliferation of the cancer cells. If you do not have that process on, then the cancer cells do not survive either. So somehow, if you can target those protein production, which is required for the cancer cell proliferation, then perhaps you can target the overall cancer pathology within the mammalian cell also. Because for the cancer cell to do its own job, you need to have this protein production. Okay, and as I said that earlier, that this happens in these three different stages of this from the gene or the DNA that we call is the process of transcription, translation, and then ultimately you are getting proteins. So we in the lab, we do this process called transcription, the initial stage from the DNA to RNA production. And obviously, there are so many other labs which are also doing from how the process of RNA to proteins are being, or the process of translation is being regulated. So those someone perhaps someday will talk. Let's just go a little bit about this process of transcription. It is shown in the next slide is that the process of transcription, it's relatively simpler if you put it in this way, is that initial stages, it starts with a process called initiation, means the transcription has to initiate. Then from there, it goes to the elongation, means that when it starts, then it has to elongate, and then it ends in the termination. So these three, very simplified way of thinking about the process of transcription, which is true for other biological processes. Anything that you start and then you keep on doing, even for machines as well. So you can think about it as a process of machine. You initiate, keep on producing, and then you terminate. Okay, and in these processes, as is shown in the next slide, is that you need multiple of different kinds of factors to do this process of transcription from the DNA to make the RNA. So the RNA would be produced, and these RNA would next be used for the subsequent steps of the protein production for the biological processes, as well as for the, let's say, for the cancer pathogenesis. Those cells would also need the protein production. So some of these factors are shown over here. You really don't need to concentrate on these factors per se, but to show that to give you a glimpse of this, that the multiple factors, they cooperatively, they act in concert to start the process of transcription, in which the protein called RNA polymerase is the key for producing RNA from the underlying DNA sequence. So in majority of the cases, there are three different RNAs are there. RNA polymerases are there. RNA polymerase 1, 2, and 3. And in majority of the proteins are being produced, that's from the mRNA, which is being transcribed by the process of transcription by this key protein called the RNA polymerase 2, as is shown in the next slide. Okay, this is the cartoon diagram of RNA polymerase 2, which is the main player in these overall transcription, the process of transcription, in which from the gene you get the RNA, and from that RNA would next be used for the process of translation to produce the protein. So in this process of transcription, you have this RNA polymerase, which is important. So the RNA polymerase, what it does is that it uses the underlying DNA sequence as a template, and it produces, using that as a template, it produces the complementary, means using that one as a template, and the template guides that what would be the RNA sequences within the RNA, and that using that template, it produces the RNA as is shown in this green picture. The green RNA is shown in this cartoon diagram, which is being used by this RNA polymerase. And these RNA polymerase mediated transcription, we can do actually, or we do very, very frequently within the lab in the test tube, that if you can purify this RNA polymerase, which is very purifiable, and then if you use the DNA sequence, that RNA polymerase starts transcribing, and then and it produces the RNA, as is shown in the next slide. Is that in these right hand side, you see that you don't have to go into these details, but as you see in the top, all these black dots or the black lines, those are the RNAs. Those are being produced by using the RNA polymerase 2 in the reaction within the lab. In the test tube, we can do all this to study the process of the regulation of this process of transcription. Okay. Now, this is a kind of overview. What I said is that going from the mammalian, the whole human body, to organ, to then from the organ to the cell, from the cell to the organelle, and from the organelle to the nucleus, from the nucleus to the chromosomes, and the chromosomes have this nucleosome basic unit, which holds the DNA sequences, and that DNA sequence gets transcribed by RNA polymerase 2 to make the mRNA, which ultimately gets translated by the process of translation to give the proteins. Okay, and which is what it's required for the overall biological processes. Okay, so this is the overview of the very simplified overview of the process of gene expression. Okay. Now the question is like, yes, I gave you a layman's idea, or in the overall process of gene expression. Now what we do, just to give a very glimpse to you guys, what we do in the lab. So we in the lab, as is shown in the next slide, is that we are working in this process of one of this process of a process of gene expression, of course, but that gene expression correlates to this disease called the leukemia or the childhood leukemia, in which what happens is that because of these, the process of the during the meiosis, one chromosome gets paired up with another chromosome, and sometimes there is a bad chromosomal translocation happens, and because of which there are sometimes this bad chromosome that give rise to the bad chromosome during the processes of the development of the childhood. And then when that bad translocation happens, it gives rise to these, in this specific case that we are talking about is this MLL fusion mediated leukemia, which, what is the underlying mechanism and all these things are pretty much not known. And we are trying to understand these through this through these processes of the gene expression. Just to give you a little bit glimpse of like what is the process of the leukemia and why does it happen. So going to the that when we talk about this process of the blood cell development, the leukemia is a process. It is also commonly known as the blood cancer. So it's the problem of the blood cell not being produced properly. So you need to have all these kinds of blood cells which are shown over here with the neutrophils, eosinophils, white blood cell, red blood cell, so on and so forth. And all these start with one common cell called the hematopoietic stem cells, and that give rise to all these types of blood cell, and that's a process through which the development happens. It's called the hematopoiesis. And during these developmental process, as is shown in the next slide, you need to have a specific set of genes on. So one case is shown in this case. Say in this case, it's a gene A is shown in this case. So during these development, what you need is that same gene expression process during the early stages of this blood cell development, you need to have that gene on. However, during the late stage of this development, where the same cell now further differentiates into other type of blood cells, then you need to have that gene off. If you don't have that gene off, so there is a problem. So the process of gene turning it on and the process of gene turning it off, both of them are key for the overall development of this blood cell. If you do not have this, then there is a problem, because the same cells which are now supposed to differentiate to the next, if you do not have that, then it doesn't go to the next step, and which is what exactly happens in the one that we are studying, as is shown in the next one. That in the process that we are studying is that in this case the genes get turned on, but because of the next, as is shown in the next slide, that because of this presence of this bad translocated protein, okay, what happens is that in the next stage, when it's supposed to be turned off, the gene set which is supposed to be turned off, it doesn't get turned off, and the gene always remain on and on. And that's not good, because you need to have those genes which were turned on earlier for the overall differentiation to go to the next stage of the development. Because of those genes are on, like you do not have those set of cells differentiated to give you the macrophage, to give you the other types of blood cell. And when that doesn't happen, then you have the leukemia, which is commonly we call the blood cancer. Okay. Now, why are we studying this? It's shown in the next slide is that the key factor in these is that is shown over here, and you don't have to really care about all these factors, just as a factor. What happens is that these factors, these fusion factors, they assist the RNA polymerase 2, or because of this novel function, they make the RNA polymerase 2 more active, and it will keep on asking the RNA polymerase 2 keep on transcribing the genes, and it assists that process. So if we know by somehow, it's shown in the next slide, that if we know by somehow how these factors are assisting the overall RNA pol 2 mediated transcription, and thereby leading it to always the genes to be remain on, then we can actually specifically target those specific processes for controlling the process of the leukemia. So if we know how actually this process is being regulated, then we can further target that specific steps. And this is the idea of studying, is that knowing what's the mechanism of these gene expression by these aberrant proteins, so that we can target it afterwards for controlling this disease. And this is the overall underlying hypothesis, overall underlying premise on which we are working with. Okay, so I gave you a very basic glimpse of what we are doing and the gene expression, and in a context that what we are doing. In fact, actually, this is also important for today's context as well, as is shown in the next slide. What happened?
The screen is unshared. Dr. Devrata, could you share your screen again?
Yeah. We are trying. Just hold on for a few seconds. Next. Yes. Okay. So as all of you know, I mean, all of us are going through this COVID-19 pandemic. And in these cases as well, the process of the understanding the process of this gene expression is also important, because when the virus goes in, it's the viral RNA that goes in, and that RNA gets transcribed by this process called transcription, which produces the viral RNA, and along with the other viral proteins, it produces the more virus particle which comes out. So, and that's the viral life cycle that goes on. So if within these process, the RNA production from by transcription, if we somehow can use some of these small molecules, then this process, then those small molecules can be used to target for targeting these viruses. In fact, actually, some of you may know one of these widely publicized small molecule or the drug called remdesivir, which actually remdesivir affects the processes of transcription from the RNA. It's called the RNA dependent RNA polymerase, which affects on that specific steps in these transcription steps to inhibit the viral replication within the body, so that we can cope up with these virus processes. And even as is shown in the next slides, even the process of gene expression is also important for the vaccine development against COVID-19. Some of you know that the Moderna mRNA vaccine is already on the horizon, and some of the developed countries have already started giving, including UK, and the US will be giving soon. So in this case, what the underlying principle is that the mRNA which encodes for the viral proteins, those mRNAs are directly being injected within the human body, and those mRNAs will be translated to produce the proteins within the human body, so that now those proteins, the body can generate an immune response against those viral proteins, so that now you are immunized against that. So that is the underlying overall working principles for this Moderna mRNA vaccine, which is already being administered within the population. So I tried to give you a kind of a glimpse of the overall process of gene expression, what we do, and then how we try to go a little bit more details about this, so that can be used for targeting specific steps for combating either different types of parasites infection, or the problem within the body with a specific steps of transcription or the gene expression that leading to the cancer formation and something, and so on and so forth. So that's the overall view that on which we are working with. And with that, I will go to the next slide. I will stop over here. All of you, please stay safe, and then pandemic is not over yet. Please keep your mask on. So with that, I will stop over here, and I'll be happy to take all the questions that you may have. Thank you very much.
Thank you, Dr. Devrata, for a wonderful presentation. And
Thank
now
you.
take some questions from the participants. So
Sure.
I request all the participants, if you have any questions, kindly post it in chat box. There are some couple of questions which I will continue with the questions if there are any doubts from your side. So the first, Sharma, she's saying, I read that PCR can help in identification of disease at an early stage. where it is curable in many cases. Can you please elaborate?
Absolutely, that is possible. And in fact, in the case of the COVID-19, the PCR actually the process of PCR is being used for early detection of the viral mRNA. When the virus goes in, it infects the body and then it injects its mRNA. So what we do is that, and some of you may know that we take the entry point where the virus is entering through the nasal passage. So we take a swab and we take the nasal passage, through the swab we take the fluid of the nasal passage and where the viral load is high, and then we get the mRNA and we extract the mRNA and we detect that mRNA by a process called the by PCR only we detect the mRNA, and then that's how we confirm that the viral infection has happened. So it's absolutely, that's the way actually we do. Yes, so your question is perfectly valid.
Right. Going to next question. Why is it necessary to use sanitizer?
Why is it
How it works? To use
why is
sanitizer.
it necessary to sanitize?
Yeah, to use sanitizer. Now in COVID time, it is said that you should sanitize yourself, wash your hand, or why
Absolutely.
of it?
Yes, I mean the name sanitizer itself it suggests that you need to, it's sanitizing means you are, it's cleaning. So sanitizer, it has some of these chemicals which kills all these organisms, because especially the key ingredient is the alcohol. So with what with if you do not sanitize, then the virus will be there on the surfaces, okay, on your palm, on your somewhere. So more and more, if you do not do that, then there will be a certain point wherein it would really be contagious, and it would, there is a chance that when you touch that your hand with your mouth, in your mouth or close to your nose, then it's going to definitely cause problems, because through that there is a chance that you would get you'd get infection. So if you intermittently, if you sanitize your hand, the virus or even other types of parasites as well will be killed, and then chances of yourself getting infected would be much lower. That's the idea of sanitization. Yes.
Okay. So, Dr. Devita, in your presentation, you mentioned about some term TSS. So, students are asking, can you elaborate upon the TSS?
Oh, TSS. Okay, TSS stands for. I'm sorry about that. I should have actually taken care. TSS stands for transcription start site. So, because the DNA is a long stretch of DNA, so you need to have a demarcation about the each gene. That this much stretch it encodes for a specific gene, and that would give to a specific protein. So every gene is marked by specific type of DNA sequences, and the process of transcription starts from one specific place, which is known as the transcription start site. It only starts from there, not anywhere within the genome. So it has to be within a specific place, and that's called the TSS, or the transcription start site.
Okay. So, the next question is from Rachna Bhatia. The gene regulation of cancer cells, will it affect our normal cells too?
Yeah, I wish I could have seen your face, Pragya, or someone, because you are asking the question that even a scientist failed to ask this question. You know, so yes, that's one of the key question that always we have, because the process of normal cell function and the process of cancer cell function, basic principle is always the same. Okay. Now, the cancer cell is always, so whenever we are targeting cancer cell, there is always a concern that that would also affect the normal cell function. If that is the case, how do we actually combat this? Now, what we do is that we try to first identify that what are those factors, what are those key factors that cancer cell completely rely on. Okay, and we try to target those specific factors, so that only cancer cell which is specific, which relies on those factors, and the normal cells may not be so much reliant on those factors. So in that case, we always kind of weigh in terms of, like, how much bad is there versus how much good is there. So if there is, let's say, 80% good is there, and sometimes we do take the bitter pill of 20%, thinking like, because there is a 80% good, so we'd go with that. So it's always the case that we always weigh in the percentage of the good of because of this versus percentage of bad. If there is always, like, let's say 80-20, then we go for 80 than ignoring 20, because at least those 80 would help. Otherwise, if those 80 are not there, then the patient wouldn't survive for, let's say, more than a year or something like that.
Right. So many students are asking again about DNA, RNA, and function of stones. So can you just summarize it in a more simplified form, maybe, because we have some students from the lower
Absolutely, absolutely. I'd be more than happy to. Okay, so the DNA is a kind of, you can think about a thread-like structure, a thread that you use at home for stitching. So you can think about the DNA as a thread-like structure in which all the properties of different molecules and everything is encoded within that thread. It says that this thread is a pink color, and it should encode these, or it should translate, or it should give rise to this specific protein, which is, say, required for Dr. Pooja's function, okay, or which is required for Pragya's function, or which is required for these. So this thread is actually you can think about a long stretch of this thread is being there with the DNA sequence. Now our body, it has to have all these threads in a very small space, because it needs to have so much of cells, so much of organs to function. So how do you keep that thread? So you keep on, packing it, and you make this compact structure, and it forms this compact structure called the chromosome. Okay, and that chromosome, when you need to express the gene, then this chromosome, this DNA have to be accessible. There are processes by which now a specific factor or the transcription factor gets access to the DNA, and through that, when it goes there, it starts the process of transcription. So that's from the thread. It has to first go to the thread, and from there, then it would make the protein. Protein means in a everyday function, the enzyme, or any other function. Even if you are eating, let's say, chicken has also a lot of proteins in it. So all these proteins are made up of the amino acids which are being encoded from this DNA, and that those ultimate production of proteins is important for the biological functions. And if you don't have that, then it's a problem. So using this DNA as a template, and then you are making the protein, so that we perform our work in a more rather without any problem day to day.
Right. So the next question is from Gift. What if cell and corona cell mix with each other?
Can you please repeat your question again? Sorry.
What if the cancer cell and corona cell mix with each other? What will happen?
Oh, a very good question. I don't think anyone has done that experiment. I would hire you in my lab to do that experiment. A cell infected with coronavirus and the cancer cell, and let's see who and with this fight, who wins. Yes, perhaps. No, not a bad idea. Why not? I have never thought about it. Yes.
Yes,
So write down the name of this student. I will contact this student. Yes.
Sure, I will give it. Then Dr. R.K. Srivastav is asking how vaccine affects to RNA polymerase to prevent transcription.
The vaccine actually is not affecting the RNA polymerase function, but rather what's happening is that with the Moderna vaccine that's there, it's actually injecting mRNA. So it's bypassing the process of transcription. Usually what happens is that when the virus gets in, then the virus injects its mRNA, and that mRNA itself further mRNAs are being produced using the host proteins, means within the cell proteins, those are there within the cell. And with that proteins, it produces from one mRNA to the other mRNA. Okay, and then that mRNA subsequently produces the viral proteins by a process called the translation. And these viral protein and the mRNA, they assemble and to form the virus again. Okay. Now, in the process of this Moderna vaccine, what's happening is that instead of using the whole attenuated viral particle or anything, because there is always a concern of the virus, getting in, and then what would happen, and then how much can it be administered, and all these issues are there. So that way, the mRNA is much more safer because you are not using the virus particle per se. You are just using the viral mRNA, which will be going in within the body, and then it would be producing the proteins of the virus. Now you are not using the entire viral mRNAs. You are using only few mRNAs. So never ever you will see viral particle is being produced within the body. So when those proteins are being produced, then the host system, immune system, recognizes those viral proteins as a foreign body, and it mounts the immune response, and that gives you subsequent protective guard against the subsequent viral infection, against the COVID-19 in this case. Yes. So it's not actually interfering with the host functions, but rather it needs the host function to produce from that mRNA to the protein, so that those viral protein will be detected afterwards by the immune system to mount the immune response.
Very well explained. So moving to the next question. The question again is from Navya. She's saying that in a lab in US, they isolated 5 genes which shows predisposition to COVID-19. The effect of vaccine on the people having those genes? I think
these people are up to date than me, perhaps. I still don't know the set of name of those set of genes. I just saw the news, and then yes, see, these are very early stages of these work, rather, in which case they are claiming that those 5 set of genes are required for the viral replications, viral these and that. But the vaccination is not going to have any effect on any of the function of these set of genes. That's for sure. Yes.
We'll now take last 2-3 questions. Use the attenuated COVID-19 as a vaccine, and in case if someone is having very low immunity, will the vaccine be still fine?
That is always a concern for while vaccinating. It's just that we always assume that, okay, 2 doses of these versus 2 doses of that should be fine across the board, but it's not always. So that's why an active monitoring is always required with the physicians, like who is doing how and all these things. But the question is absolutely valid, is that the immune response for those who have the weak immunity versus the strong immunity would be different. So who can mount how much, and then who can take how much doses and all these things, those are absolutely variable and cannot be generalized in population wise, but that concern is always there. But has to be properly, after vaccine administration, has to be properly, always have to be under supervision of the physicians to see the response. Yes.
So I'm taking the last question now. What is the role of T cells in fight against cancer?
What is the role of T cells in fight against cancer? So these are one of this field called it's called the cancer immunotherapy is coming up, in which the T cells are being used, engineered in such a way so that it detects the cancer cell. Now these are all engineered T cells. Okay, it detects the cancer cell, and then the function of the T cell is to kill the parasites and kill others. So that way it has been engineered in such way so that it can detect those bad cancer cell and it can kill them. So this T cell, under normal condition, it just fights against all these infections of the parasitic and any other infections. But in this case, we are slowly moving towards the way in which the T cells would recognize the bad cancer cell and would kill them. And if it is so, then it would be better, because then in that case, we don't have to bring any other foreign material within the body. So just by isolating some T cells, engineering it against the cancer type of cancer that we are dealing with, and can be administered back to the patients, and then patient response against the foreign body wouldn't be there, and the overall efficacy would be much more. So that way, normal function of T cell is to give the immunity against the infection, common infection, and others. And it's being designed, engineered in such a way so that it can also fight against the cancer cells as well.
Right. Prita, there is one question I got from YouTube. I forgot to check over there. So I would request to answer this question as well. This question is from Srihari. He's asking, can the editing of gene in order to prevent disease could cause another disease?
See, we always hope that's not. Okay, because when you are editing it, you use a process, and then you are thinking that just by editing that specific gene wouldn't be having any effect subsequently, but that concern is always there. But then always checks and balances are there in terms of making sure that 20 years down the road after editing, that it doesn't cause much of an issue and all those things. But there are chances, but those are very minimal chances, rather, I would say, because we always target the one in which we try to correct it if there is any problem by editing. Not try to make it, not try to make it a kind of monster or Frankenstein so that there will be a problem afterwards. So we always try to do basic minimal editing that's required so that whatever the problems are there, that can be corrected. So that's the way perhaps we deal with it.
Right. So we're almost done with the questions. There are still questions coming which are almost similar. You already answered. And many questions are related to what if vaccine is given to a cancer patient? What if these, So I think that's a good idea if to study how this vaccine could work with a cancer cell or a cancer patient.
Yeah. It's a very early stage. We don't
Yeah.
we really don't know so much about this SARS-CoV-2. So we are knowing more and more, and then perhaps few years down the road we can merge both these COVID biology as well as the cancer cell to know more about how things would work out. Yes.
Okay, so I think now we should conclude the session. So thank you very much, Dr. Devrata, for a very wonderful session. And I see answering the questions, all the queries from the student side. And the students are requesting for your email address. So I hope it's okay if I share your email address.
Absolutely not a problem at all. Yeah.
So thank you very much for your time and for very exciting session. And we look forward to have you maybe in future for other sessions under this program.
Yep.
So I'm sure all students have learned a lot out of this session, and they could rethink, they could new ideas, applying certain new ideas out of this talk today. So thank you very much, and I also thank all our students and putting up very nice questions. So stay safe, take care, goodbye.
Thank you very much, everyone. Yeah, bye bye.